Hey guys, it's Abby. Exponential technology surrounds us every day, making our lives easier, healthier, and safer. One of the most up-and-coming being gene editing. Gene editing can make horses into unicorns, bring back the woolly mammoth, and design your dream baby. Before we get into exactly what gene editing is, let's go over the basics. The human genome has 4 types of bases found in DNA: adenine, thymine, guanine, and cytosine. Adenine pairs with thymine and cytosine pairs with guanine. The genome is comprised of over 3 billion of these base pairs in 2 intertwining strands of DNA. The sequence of these bases encodes genes. DNA is transcribed into RNA, which is translated into protein. Misspellings of even a single letter of a gene, known as point mutations, can yield proteins that are dysfunctional or missing altogether, this causing disease. Gene editing is a type of genetic engineering in which DNA is inserted, deleted, modified, or replaced in the DNA of a living organism, and this can correct the misspellings in the genome. There are many companies utilizing the power of gene editing to combat genetic diseases, but today I'm going to focus on Beam Therapeutics. Beam Therapeutics, launched in 2018 by Liu Jing and J. Keith Jung, is pioneering the use of CRISPR-based editing to develop a broad portfolio of advanced genetic medicines. But hey, what even is base editing? I'm glad you asked. Base editing is a new genome editing technology that enables direct and irreversible conversion of a DNA base into another at a targeted genomic locus, or a specific location on a chromosome. Base editors have 2 principal components fused together to make one single protein. First, a CRISPR protein bound to a guide RNA that leverages the established DNA targeting ability of CRISPR, but modified such that they do not cause a double-stranded break. And second, a base editing enzyme such as deaminase, which carries out the desired chemical modification of the target DNA base. When introduced into a cell, the CRISPR targets the desired genomic location by recognizing a complementary section on the DNA to the section encoded in the guide RNA. The deaminase then makes the desired edit to a target base in the editing window. By changing the guide RNA portion of the CRISPR protein, they can quickly and precisely retarget base editors to different genomic locations based on their gene sequences. By changing the deaminase, we can control which base is edited. Base editors have several advantages over existing gene editing approaches. 1. It creates a precise, predictable, and efficient genetic outcome at a targeted sequence. 2. It edits at high efficiency without the need for template-based homology-directed repair. And 3. It avoids unwanted consequences of double-stranded DNA breaks, such as frequent insertions, deletions, or even larger-scale genomic rearrangements. Beam Therapeutics hopes to provide lifelong cures to patients suffering from serious diseases utilizing base editing. For example, one of their projects focuses on treating sickle cell anemia through complementary base editing. The study will be linked in the description below. Thank you so much for watching. Please like and subscribe for future videos just like this one. See you next time.